data, a day to explore digitization in the collection, and possibly to increase access. So before we start, let's start with land acknowledgements. The natural history collection have historically and currently benefit from colonialism. Specimens shipped on board slave ships to glorify empires and to promote exploitation and stolen objects and so much more. The time acknowledgement is long overdue, and we must acknowledge that through our science, as we continue our science, and we have to continue to impact Indigenous people. We are all based in different locations, from Los Angeles to Gainesville, Florida, and across the globe. These sites are traditional territories for the Gabrielone Tongass, the Seminole, and the Tomoka, and many other land tribes that we're not acknowledging. So Black and Natural History Week has been organized by numerous people. There's our founder, Adania Fleming, and many other people. We've been meeting on bi-weekly to just start this organization. However, we've also been meeting for specific days that we've been participating in. So today's day 5, our lead is Hank Bart, and there was numerous other people participating to make this day happen. So ground rules when participating. Participants' videos and microphones will automatically be turned off upon entry. Participants can choose to have their videos on or off. Just keep in mind we're recording, so if you don't want your face shown, turn off your video. We ask that all participants remain muted when the active speaker is talking, and again, note that the session will be recorded. Some ground rules. We will not be using the raise hand function. If a participant wishes to speak or ask a question, they can send a question mark to the chat monitor. Our chat monitor today is Brianna. And the chat monitor will then send the question in a doc, and then I will get to the questions at the end of everyone's presentations. Zoom basics. Not everyone has been using Zoom for the past year, 2 years. I don't know how long the pandemic has been going on now at this point, but at the very bottom of the left screen, you'll see the mute and the video function. And so let me see if it'll let me go to Zoom. It will not. On the bottom panel, there'll be other functions such as the chat. That's where you can privately message the chat monitor. There's also other functions. Hopefully you'll be able to access as you need. When it's time for you to speak, you can just click unmute, and then to find the chat monitor, again, go to the chat and specifically request the chat monitor. Code of conduct. The registration and participation in Black and NHM is a sponsored event and represents an agreement to abide by the Black and NHM Code of Conduct. Please note that the following expectations can be seen on iDigTrio. The link is below. Adania was a part of this org, and we're going to follow this that she previously made. Individuals attending Black and NHM's virtual events have the right to an environment free of discrimination, bullying, inappropriate behavior, and harassment. These behaviors will not be tolerated at Black and NHM activities, including the virtual events. There's prohibited conduct that you cannot, that we're classifying as misconduct, so discrimination on the basis of protected traits, harassment on the basis of protected traits, sexual harassment, retaliation, bullying, and other unaccepted behaviors. If you want to report any violations, please contact the moderator, me, of the event in and/or virtual events. So if it didn't happen during an event, but on Twitter, you can go ahead and contact blackandnhm@gmail.com. Incidents of inappropriate behavior during any virtual event will be taken seriously. Violations may result in being removed from the virtual event and/or being prohibited from attending. So let's go ahead and talk to our panelists today. We'll have 4 panelists, Hank, Lisa, Philip, and Corey. So today we're going to start off with Hank. Again, leave all questions to the end. All questions will be left to the end, and that's when we'll answer questions.
Thank you, Sharon. I know you took it away.
Yeah, I got it. Yeah, I had to unmute myself. Thank you.
Gotcha.
And thanks very much to Hadil for moderating, excellent job. To Brianna for serving as the, I'm sorry, Brianna, what'd you call your position again?
Chat monitor for
Chat
today.
monitor, yeah, vital. Chat monitor is going to help us see who the questions are coming from. And to our fearless leader, Dania, for being the bouncer. I love that. Okay, so we're going to hear presentations by some excellent presenters. I'll lead off. Then I'm going to pass the baton to Lisa White, Director of Education of the University of California Museum of Paleontology. Next up will be Philip Skipwith, who is presently an assistant professor of biology at the University of Kentucky. And then lastly, we're going to pass the microphone to Corey Evans, assistant professor of biosciences at Rice University. So let's get into it.
Hank, before you start, do you want to, oh, there you go.
Yeah. All right, so we're going to,
wait, Hank, do you want to present your whole slide? Because we're seeing the slide set up. Do you want to?
oh, I'm sorry. Let me just one more click. One more click, and then I should have it. How's that? You see it?
No, it's still.
I think because I have two screens. Hold
Can
on.
you
I'm going to stop share, and I'm going to get
Okay.
the other screen.
And while Hank's doing that, the transcript is enabled. So if you would like to have the caption during this panel, you can go ahead and enable that on your screen. It's in the bottom panel.
Do you see the full screen now?
Yep, it's perfect. Good to go.
Oh, good. Okay, so we've got some important topics to consider today. Obviously, the most important one is digitization, which is globalizing access to data and images of specimens and natural history collections. You're going to hear presentations that cover a variety of different approaches to digitizing natural history specimens today. And then secondly, this is not a subject we're going to cover in depth during the presentations you will hear, but we are going to address this issue of, the impact of colonization on the distribution of natural history specimens around the globe, and efforts now to decolonize natural history collections through processes like repatriating the specimens back to the countries of origins. So hopefully we can hold discussion around this topic after we finish our presentations. But the hypothesis that I'd like us to talk about is whether or not these digitized versions of these cultural artifacts or suffice as a repatriation of those objects, or maybe might substitute as an effort to repatriate those objects, or can we instead repatriate the objects to the countries of origin and just keep the analogs, the duplicates of those in our museum. So that's something hopefully we'll get into in the talk as well. Okay, so now, there we go. All right, so I'm going to talk to you about 2D and 3D digitization of fish specimens for a project involving machine learning-based trait discovery. And this is a project involves a lot of institutions, a lot of collaborations funded by the National Science Foundation. The project is called Biology Guided Neural Networks. It was funded in 2019, an outgrowth of an NSF Harnessing Data Revolution Ideas Lab project. This is a really fun activity. If you have had the opportunity to participate in one, I highly recommend it. What the project is doing is applying machine learning methods to large volumes of 2D fish specimen images to see if we can train machines to discover previously undiscovered traits. And this will allow us to complete accurate trait distributions and new trait ontological relationships. And if you saw in the first slide, one of the collaborators is Dr. Paula Maybe, who was instrumental in building this really great platform for understanding ontological trait relationships called Phenoscapes. So what we're doing is we're segmenting these 2D images of fish specimens humans are doing that so that we can use those as training data sets to train algorithms how to extract the same kind of trait information from the specimens. We're incorporating information from phylogeny showing relationships of the different species we're working with, and these ontologies, these trait ontology terms, to improve the predictions of the machines in identifying these traits for us. So, and if these efforts are successful, all the information is fed into the neural network. It spits out at the result. We look to see if the neural network's predictions are accurate in terms of what those traits are. So what we're doing is we're starting off with just plain images of 2D images of specimens. We're doing things like landmarking the images, segmenting features like the heads, the different fins, the eye, the mouth, etc. And then, we develop training sets of these images, and we feed those training sets into the neural networks to produce the results. So the question is, are 2D images adequate for representing the features of specimens? They do reveal a lot of information about fish specimens, but they don't really capture the true 3D form of the specimen. The width dimension of specimens is not part of a 2D image. And the relationships of any structure that is lateral to the mid-sagittal plane of the specimen is not captured by that 2D images. We use a left side lateral view of the specimen to do these 2D, to extract this 2D data. Fish are bilaterally symmetrical, so your assumption might be that the two sides of the fish are the same, but that's not necessarily true. Sometimes the two sides can not be perfectly symmetrical because of a phenomenon called fluctuating asymmetry. So this brings up the question, can we produce 3D reconstructions of specimens from 2D images? And by 3D reconstructions, I mean 3D surface reconstructions. Well, it turns out you can do this computationally by stitching multiple photographs of a specimen together using a process called photogrammetry. And in fact, this is a method that's been around for a long time, and you can actually do it manually, like I did for this specimen of a stone roller, which is shown in the middle of this slide. So what I did was I manually took 243 images of the specimen. I had it propped up on a support that was painted with green screen paint, so I could remove that support image from the photographs, from the images. And then I took 243 images from all angles around the specimen. And then I fed those images into a computer, and I produced this model of the specimen. And you can see that this is not a bad reconstruction, 3D reconstruction of that specimen. I can see features like the scales of the specimen in very good detail. I can see the breeding tubercles that the specimen happens to have on it. The fins are a little ragged. However, the biggest problem with using this manual approach is that it takes a long time. It took me about an hour to take all those pictures, moving around all the different angles and whatnot. And the specimen got kind of dried out as a consequence. So, oops, wrong slide. No, there it is. So thanks to a grant from the Institute of Museum and Library Services a couple of years ago, we received a grant to build a prototype of a photogrammetric imaging system that we call COPUS. And you can read about COPUS at the website that's shown below. But I just want to show you an actual demo of how it works. So there are 6 cameras that are mounted on supports. And these cameras rotate around in the upper plane and the lower plane of the instrument, and they pause and they take pictures. And so 6 cameras reduce the amount of work by a single camera by to 1/6 of the time it would take. But actually these cameras can find their positions, take their photographs faster than I could. And so they cut down the time, which took an hour to image a specimen from to about 5 minutes. So we were able to produce capture enough images to produce a photogrammetric reconstruction in just 5 minutes time. So here's another illustration of a photogrammetric reconstruction of the jaws of a sawfish. And this one was produced with Copus. And you can see it's the same kind of quality. It's a different kind of specimen, same kind of quality, but it was produced very quickly. So Copus is a very good instrument for producing these, oops. I pulled out my headphones, for producing 3D models of fish specimens or shark jaws or anything else. And you can do this fairly quickly. It turns out we did not yet get a grant to use this technology. However, we have a newly funded project, a 5-year project to extend the work we're doing in biology guided neural networks to fish and all other kinds of animals. It's called the Imagomics Institute. And we're also going to be doing these photogrammetric reconstructions for this project. Just a little footnote, I'm looking for a computer savvy postdoc and a doctoral student to work on fish-related aspects of the Imagomics project at Tulane. If you're interested, please email me. My email address is there. And with that, I'm going to stop. Thank you all for your attention and pass the baton to Dr. Lisa White.
Great. Hi, everyone. I'm going to go ahead and share my screen and get started. And I'm going to highlight what happens to those digitized products when we're thinking about broader audiences, the pre-college community, or even the community college community. What are some ways we can share the products of this work with others? So the icons that you see there are for some different projects that I'll mention that I direct at the University of California Museum of Paleontology. I'll have a number of URLs on the slides. I'll also have some QR codes at the end, so you can feel free to screenshot or take photos if you want to access those websites. Now, I should say, by way of background, I'm a micropaleontologist by training. So I specialize in fossil diatoms, as you see there on the lower left, when I was more research active. So a lot of my duties now are in education and outreach, science communication. But in my research intense days, I would participate in International Ocean Discovery Program cruises, be one of the micropaleontologists on board, dating the core. I've also done a fair amount of field work all around the Pacific Rim, close to home, looking at the Monterey Formation here in California and diatomite quarries. And as I've shifted the last 9 years since I've been at the Museum of Paleontology at Berkeley to focus on education and outreach, I'm able to bring educators and graduate students out when I am able to go out on ships. And of course, post-pandemic, hope to return to being out at sea again. But I want to mention that the University of California Museum of Paleontology, UCMP, we've had an online presence for a long time. We were one of the first 100 websites really anywhere in the world when the web was in its infancy. And so for decades now, we've been sharing a whole range of resources with the general public and K through 12 audiences. And so it certainly makes sense now as we're entering a time when we're digitizing more specimen records and we're sharing databases that we make sure there are products appropriate for the general public and for K through 12 audiences. And so the staff, faculty, and certainly the graduate students at UCMP have all been instrumental in this work. And we've benefited from a grant from the NSF as part of a Thematics Collections Network grant affiliated with iDigBio, which is, of course, housed at the University of Florida. And with our particular Thematic Collections Network project, it focused on the record of Cenozoic invertebrate specimens throughout the West Coast of the US or the Eastern Pacific. And so with more than 8 other museum partners, we digitized over 1.5 million specimen records, photographed hundreds of specimens, had increased a very robust georeferenced database now with localities. And it was important throughout this work to update taxonomic records and fundamentally at the end of the day, just share what information we have and make sure all of our information agrees. And as the individual working with the team on this product for the broader impact materials, the education outreach materials, we decided to focus on virtual field experiences. So really connecting the places where we make that primary data collection. And then what happens once those specimens are collected as we then digitize them and make them part of our database record. So this EPIC product, Eastern Pacific Invertebrate Communities of the Cenozoic, included all of these museum partners and the millions of records, specimen records at our combined museums was certainly worthy of supporting and communicating the information about what those records glean with the public. So this focus on the field experiences and how to really transfer and make those connections between those primary data collections in the field, setting that context, and then what we do with the specimens once they get to the museum. So we made a series of modules using Esri story maps as our primary platform, and with the Paleontological Research Institution in Ithaca. So we took the lead on this arm of the EPIC project. And so really using a range of tools, we used a high number of Google Earth images, gigapan photography, really putting it together with a number of specimen records and really supporting ways to think and learn about geoscience in the context of paleontology that adds value to educators who are looking for resources to align with the next generation science standards, ability to really provide context and connection to why and how fossils are important. And so along the way, we really continue to ask ourselves, why is this particular format effective when there are plenty of ways to share science? And we see that in the iDig BioNetwork from materials that are very citizen science friendly, different kinds of crowdsourcing opportunities where people help with collections, but we were very excited to be able to share from the field perspective how this is all connected. And so here's just an example when we're in the field of capturing images with gigapan photography, where we have the ability through billions of pixels of resolution to capture all this detail. And so this was taken some meters back from the cliff, but with a series of images of that cliff, we were able to really detail what you see. And so when one takes this journey in these different modules for the virtual field trips, there's opportunities to scan and zoom in and out. We want the user to see what we see as researchers, but there is a lot of different levels of scaffolding so folks can take advantage of this. And there's video assets, there's an illustrated glossary, there are opportunities to learn more about these field areas. And so clearly we had some fun while we were doing this as well. And I'll just spend the last couple of minutes talking about how other digitized resources have made their way into other programs at the UCMP. And several years ago, we initiated a program with community college students in mind, so advancing community college education and student success. When we started this program pre-pandemic, it was an in-person opportunity for community college classrooms in the Bay Area to come to the Berkeley campus, and UCMP graduate students would design a 3-hour lab that would support knowledge about paleontology. But as we transitioned to remote learning last year, the graduate student leads for this program included a number of digital resources as part of virtual remote labs intended for this 2-year college audience. So they can be used synchronously or asynchronously, but it was our intent to really capture all of the kinds of information and material that's available in these digital databases, including some of the ones that you see here. So working even beyond the EPIC resources, but tapping the iDig Bio Network, as well as the Digital Atlas of Ancient Life. These have all made their way into these resources intended for the 2-year college audience, although we now have users from the 4-year college audience as well. So I'll move along, but again, feel free to screen capture any of this and scan the QR code so you can go to this article. I'm also a co-PI on the Biodiversity Literacy for Undergraduate Education grant, the Blue Data Project. So we've been publishing this work with them in mind and with the Q's Hub folks. And this last slide just shows some of the available labs that we have community college partnerships with, but they are also very applicable to 4-year college audiences. And it's our intent, again, to really bring these high quality images into a teachable format in a context that's exciting for our users. So thanks so much, and glad to be able to present that.
So I guess it's my turn. Hi, I'm Philip Skipwith. I'm at the University of Kentucky. I go by Skip. So I just started my position in the August of 2020. So I hope everybody can see my screen. Oh, my screen's acting up now. So bear with me. My research focuses primarily on, like, a molecular biologist by training, but my research focuses now mostly on the comparative phylogenetics of squamate reptiles. And one of the things that really interests me is looking at squamates represent one of the most diverse groups, or the most diverse groups in terms of species of amniote vertebrates in the world, and they run a huge gamut in phenotypes. And people typically focus on the external morphology, but typically to examine the internal morphology of these taxa requires destructive sampling. And as a museum scientist by training, and I did my PhD at the same institution as Lisa. I was at Berkeley, and then I went and did a postdoc at the American Museum of Natural History in Manhattan. Destructive sampling pains my heart to have to do that. So one of the things I wanted to be able to do is to study the osteology of these taxa non-destructively. And one of the neatest tools that we have at our disposal now is CT scanning. So Hank was talking about using photogrammetry to study external morphology, which in some cases may be preferable for large data sets, depending on your funding source or the facilities you have available to you at your institution. Or for many institutions, particularly museums, now have readily accessible CT scanning facilities. I'm just showing some highlights of the external morphological diversity across squamate reptiles. But when I was at the American Museum of Natural History, I started a project using CT scanning to examine the internal morphology of a particular group of snakes. I work on canophidian snakes and geckos for my research. And now CT scanners are readily available at the American Museum of Natural History, the University of Michigan, University of Florida in Gainesville. I see Dave Blackburn's on this call. And now we actually have one at my institution, the American Museum of Natural History, University of Kentucky, which ironically has no collections. So it's mainly used for engineering. So I've gone from relying extensively on in-house collections to having to beg people to send me their specimens to the middle of nowhere. So the American Museum of Natural History actually had a GE Phtomics, which is one of the larger, heavier units with a more powerful X-ray beam, and I'm now using a thermoheliscan, which is a much smaller unit by half the weight, so it weighs more than an SUV. That's typically better for dealing with small specimens, typically used for engineering for microscale analysis. And to use, I had shown some of the external morphology of scories before, but we're now actually able to use X-ray computed tomography to examine the internal morphology for segmentation and examining the specific elements non-destructively, which is extremely helpful. So you can boost the actual number of specimens that are used for internal morphological analyses that will otherwise be unavailable due to destructive sampling. And in my research specifically, I'm looking at cranial diversification across snakes and geckos, but also looking at the development of fangs and the development of modularity across the squamate skull, and combining that with the genomic analyses. And when I was at the American Museum of Natural History, you know, I had a strong component of outreach. I was actually teaching undergraduates how not undergraduates, these are actually high school students, to how to actually segment CT data. But one of the major issues when you're dealing with huge amounts of CT data or any other high resolution image data, it's the same issue you have with dealing with genomic data sets, is where do you put these data? Where do they end up? You can't store them locally easily. And it makes it very hard to actually share those data with other researchers and with the public. So I did bio, Lisa had talked briefly about it, has another initiative led by Duke, with the PI at Duke University. And I'll preface this with saying that I am not a lead on this project. I'm merely a contributor, but it has definitely made my research easier. And for people who do not have in-house CT scanners, being able to outsource your specimens, or having museum loans scanned in-house at another institution, being able to retrieve those scan data is made easier by this iDig bio initiative called Morphosource. So I'm just going to open up this link here, and hopefully you guys can you guys still see my screen here? So Morphosource is great because it is this huge repository of CT scans and other photogrammetry image data of specimens from various institutions. And for me, it is particularly appealing, and I'm really glad this exists, because as a researcher, you have your own dashboard that allows you to have multiple projects that allow you to collaborate with different researchers, which is incredibly useful, but allows you to store your CT scan. Say for a time that I didn't have, like, our CT scanner at the AMNH actually broke. These machines were super expensive, but incredibly unreliable in terms of maintenance, and they're a pain to maintain. So I was sending my specimens to the Florida Museum and having Ed Stanley actually scan a bunch of specimens for me. So he was actually able to share these scans with me, even though these data sets, it was much cheaper to have these specimens stored digitally than to have that, like, a hard drive set back and forth between New York and Florida. And this is a great resource for researchers, because now, instead of having these huge data sets that are composed of unreconstructed scans, you can just basically take your superimposed TIFF stacks, your reconstructed TIFF stacks, that can now be downloaded. And as a researcher, this is great because I can share this with my collaborators, but then you can put basically a moratorium on this data set until it's published, right? So it is available, but if somebody sees that I have scanned, so I have a folder just for my AMNH scans, if someone wants access to these data, they can contact you directly and ask if they can have these data. They can't just download it at their own discretion. And this is great because it allows you to protect your data set, but also potentially open up opportunity avenues for collaboration between researchers that you actually otherwise wouldn't know. So I have dozens of scans that are just sitting on here that I'm currently working on, that people have already contacted me about, and it allows researchers to know what other institutions are also working on across data sets. So again, I'm not a lead on this project, but I have found it to be a great resource. One of the other resources that was relying when CT scanning was becoming more readily available for non-medical biology was Digimorph, which is now kind of defunct. And that was through UT Austin, and there was basically no way to get those data. So this is just a great resource that I think is incredibly collegial and is a great avenue for people who otherwise wouldn't have the computational or technological resources to have access to for their own research. So with that, I'm going to end, but with I'm going to go back to my slide here. I'm trying to get back to well, anyway, you can't see my slides, but I'm going to end with the shameless plug that I am currently looking for graduate students who are interested in comparative phylogenetics and phylogenomics who would love to live in Kentucky and work on really cool squamate reptiles. So if you know anybody, please tell them about my research. So I'm going to end my share.
Thank you, Phil, Skip. And so now, Corey, the stage is yours to present on your fine research.
Cool. Awesome. All right, let me get this going here. Cool. All right. So my name is Corey Evans. I'm an assistant professor at Rice University, and I study the morphological evolution of fishes. So I basically do a lot of what Skip does in terms of geometric morphometrics, modularity, but I don't study squamates. I study the better class of vertebrates, which are the rayfish fishes. Sorry, Skip. In general, collections have always been a really, really important aspect of my research. I got my start working on neotropical electric fishes at the University of Louisiana Lafayette. And back then, in those days, in the 2013 days, instead of using micro CT scanning, I was doing a ton of clearing and staining and radiographing. And it became almost immediately apparent how painful this kind of destructive sampling was. For those of you who don't know what clearing and staining is, basically we make the skin of an animal transparent and we stain the bones red and the cartilage blue. And while it makes some really, really cool images, we destroy a lot about that fish. I remember actually for like the first, I think, 6 months of my PhD, I actually didn't know where fish muscles went because we cleared and stained straight through them. Like I legit had to like skin a fish before I could like figure out, you know, what the non-bony parts were. So destructive sampling was kind of a bummer. And on top of it being a bummer, museum collections were not really into you clearing and staining holotypes or, you know, clearing and staining lots that only had one specimen in them. So I rapidly, and I've always kind of been looking for alternatives. So for the rest of my dissertation, for the most part, I was working on radiographing. So I was radiographing these electric fishes, and I was getting these nice 2D, you know, like doctor images where it's just like a fish skeleton with very little context. And towards the end of my PhD, I figured, you know, maybe I could do a little better than that. So Adam Summers had gotten that grant for Scan All Fishes, which basically allowed people to travel up to Friday Harbor in Washington and scan fishes for free if you were willing to endure a bus trip across the country and then sail across the sea. You could scan all the fishes you wanted, and I was really into that. So that happened at about 2017 for me. So at about 2017, I switched over to scanning fishes in 3D. And that really, I think that really kind of changed the trajectory of my career for me, because now I was able to non-destructively sample some really, really incredible specimens. So the photo I showed you before was me standing in the Bell Museum. That's right in my postdoc at the University of Minnesota. I had a grand time at the Bell Museum, and one of my favorite things was I was able to go through these really, really old collections, things like David Stark Jordan collections, and just pluck out fishes from the jar and CT scan them, and then put them in, and put them back in their jars, no harm, no foul. So along the way, I learned a lot about kind of scanning some of these old fishes. At first, I was scanning them just to collect shape data, and I would, scan them, turn them into a 3D surface, wham, bam, that's it, move on to the next fish. But when I started getting into these really, really old specimens, other things started kind of like popping out. So here, this is a Acanthurus triastegus, or a convict tang, right? And this fish was actually collected in 1930. And the scan looks like, this fish was caught yesterday. What's really, really interesting about this fish is that it was literally collected before like Black people had rights, right? And this is like a common theme if you're paying attention. This is a common theme in a lot of museum collections, especially in the older collections. You're encountering specimens that were collected by these white men who were sailing the world while my ancestors were either working in fields or barred from basically participating in academia. So this kind of became a running trend for me. And now, thanks to the help of Andrew Simons, the curator at the Dunn Museum, I've basically been running around trying to find the oldest fishes that I can find and CT scan them, because I want to make a really nice kind of composite of what was collected before we people of color were able to collect it. So this next fish is right now, this is the oldest fish that I've ever scanned. This is a luvar. So it's this like really rare, weird pelagic surgeonfish. And this specimen was collected in 1869. So I don't remember I don't remember how long it took for the slaves in Texas to find out they were free. But it's it's ballpark, right? So this is a really, really old specimen, and I got this from the Academy of Sciences, which is kind of my home collection in Philadelphia. And if you look at the actual specimen, it's like old and like really floppy and kind of degraded, but this scan really came out beautiful. So in general, CT scanning has really kind of changed my career in this way. However, CT scanning has also shown me and created a whole bunch of different avenues of research that I was literally completely uninterested in beforehand. So getting my start as a marine biology student in South Florida at Nova Southeastern University, I knew about tongue-eating isopods, and I was really keen on avoiding them at all costs. However, if you're shooting X-rays to a bunch of fish heads, you are going to find really, really terrible things like tongue-eating isopods. So occasionally I run into these really gross things, and I'll share them on Twitter. And whether I like it or not, this is becoming an active wing of my research because now like news outlets I just got off the phone with the Houston Chronicle like 2 hours ago because somebody in Galveston caught a fish with a tongue-eating louse in its mouth, and it's just never going to end. My email is like filled with like random strangers who send me these really like disgusting images of parasitized fish mouths, and that's just who I am now, right? And so I'm just going to lean into that. But in general, CT scanning and really high resolution imaging of these animals has really added entirely new wings to my research that were previously not there. These days, I'm really focused on I try to focus on skull shape evolution of fishes and other things sometimes pull me away. So here we have a wrasse with most of the bones segmented out. I've kind of cleared out the opercle and things like that. Because like Skip, I'm really interested in studying integration and modularity across the skull. And so we've been working really closely with researchers at University of Chicago to come up with methods to analyze skull shape in really complex structures. So one of the issues with the fish skull and the snake skull in general is that the skull is highly kinetic. That means it's really, really mobile. So when a fish opens its mouth, like 10 bones move at once. And when they close it, multiple bones move at once as well. So when you're dealing with museum specimens, one of the challenges is that fishes are generally not preserved in a like standard position. If you go through a mammal collection, all the preps are really, really nice. You have the little neurocranium, and then you have the mandible set off to the side, and then you have a little fur. If you are running through a fish collection, it's just boys in jars. And they're like bent up. Sometimes their mouths are open. Sometimes they're gasping for air when you collect them. So you get all these really, you get these things in non-standardized positions. And this creates problems if you're trying to do geometric morphometrics. So because each fish will look like its mouth is closed and its mouth is open, and that'll change the actual shape that you're calculating. So we figured out ways, we published a new method called the local superimposition that allows us to basically mathematically standardize the position of all these different elements in the skull, and it's really changed the game for a lot of the research that we did. So some of the research that I have been able to do with these micro CT scans is analyze patterns of variation across a ton of different species. So because the sampling is non-destructive, I don't have to wait like 6 months for a big fish to clear and stain. In grad school, I think I might have cleared and stained like 80 fish, and it took a whole summer, and I had just batches of trypsin and glycerin everywhere, and it was a nightmare. And my advisor, fortunately, he was out of town for like the summer, but he came back, and it was not great. These days, we don't have those kinds of problems. So this is the analysis of skull shape, looking at caringarians, which are a gigantic clade that includes flatfishes, billfishes, barracuda, you name it. And we were just focusing on the nerve cranium for the study. And this study incorporated about 110 species. And it was a really fun experience because it really only took 2 trips to collect all this data. I spent about a week at the University of Michigan using their scanner, and then I went to Friday Harbor for the summer and just scanned every flatfish I could get my hands on. And in those 2 trips, within a year or two, we had all the data that we needed for this project, which is pretty wild. And then Morphosource helped out a little bit as well, where we could pick out little species that we didn't already have. And with all that data, we were able to analyze patterns of skull shape variation, and even patterns of skull shape evolution across an entire clade. So this is the scale that we're at using these non-destructive sampling methods, where we can look at like really rare specimens that might only be represented by a few specimens kind of globally. I'm actually still on the hunt, shameless plug here, not for a grad student, but for a roosterfish. If somebody can get me a roosterfish that can fit in a CT scanner, that would be really great. I actually have my own scanner here at Rice in my lab. We just got it, and it's great. I think it holds up to 30 centimeters in length and 350 centimeters in diameter. So if somebody in this room can find me a roosterfish, that would be great. In general, though, back to the work. Using these non-destructive sampling methods, we can get kind of a clearer picture and a more broad scale picture more quickly of patterns of morphological evolution. Furthermore, using these same methods, we can even expand like globally, right? So here is an analysis I have in prep, just looking at the rates of skull shape evolution across races from a global perspective, and looking at the relationship between rates of evolution and latitude. So these are just some examples of what we can do with these really high resolution, low impact, if you will, data collection methods. And it's really exciting, especially now we have a better handle on how to analyze it and how to standardize things. It's really shaking out to be a really exciting frontier. And that's all I got.
All right, thank you guys. Oh,
That's right. Go ahead.
you're good. I just want to say thank you so much to Hank, Lisa, Philip, and Corey for presenting today. I'm switching over from fish molecular phylogenetics to more fossil data and morphology. So I came here to learn for myself and for my own research, but I'm sure everyone got something out of this. In addition, I want to thank Adenia and Brianna for being a bouncer and monitors, and I believe Lindsay for also live tweeting. I think that there's some questions. I'm gonna assume we're gonna go past the 1 hour mark and go past 5 o'clock. If anyone has any immediate questions, we can go to those first. If not, I'll go ahead and go to the questions.
Yeah, and I for one am willing to stay on a little bit longer. Are the other panelists able to do that?
I can stay on for approximately 4 more minutes, and I have to meet with Mike Sandell about,
Okay, understood.
about
Well, thank you in advance. Maybe we could pose a question for Corey if he's got to leave soon.
And if folks recall, in terms of the logistics, if you want, everybody's muted. If you want to be unmuted, you have to send the chat monitor a question mark. If you don't want to ask the question, but you have a question, you can send them the question, and then the moderator will relay that.
All right, It seems right now the questions are general questions. So I'll go ahead and ask those. If I see any Corey specific questions, we'll switch over, I guess. All right. What barriers, technology, or social experiences have you personally came across, or have you observed these through digital resources? I'm not sure. I think what the question is trying to say is the barriers you've maybe have. Oh, someone. Okay. The barriers you've came across, do you think this is more socially, like in person, or do you think a lot of these like barriers that you've came across are through digital resources or gaining information, maybe through the web or through books, accesses that are not maybe so much socially stuff, but more gaining information?
Anybody want to take a stab at that?
I think
Instead of-
I think so. I think some barriers usually boil down to researchers who may not always be so keen to, share their scans or anything for particular reasons, because maybe they're still working on it. But sometimes they've been working on it for like 15 years, and you just really need that fish, and you don't want to sail across the ocean and catch it yourself. So sometimes these are some of the barriers that I generally encounter. Or sometimes, especially with like these larger scanning initiatives, sometimes like an entire fish will be scanned, and it was scanned to be, in a repository. It was scanned to just kind of be a representative thing. But maybe you need a particular aspect of morphology that it might not be high res enough or something like that. So sometimes you have to do it again.
Yeah, it sounds like in Skip's presentation that these platforms like Morphosource have sort of made the work with specimens like this and this kind of technology more collaborative, because people know what you're working on. They can express any immediate interest in it. You might feel pressure to do something with it yourself before they do, which is a good incentive, for working on things. So is that the case, would you say, Skip?
Yeah, I mean, in my experience, I mean, it's great to be able to just see, like, I'll piggyback off what Corey said too. It's great because those sorts of, it's like GenBake, right? Like sometimes it's just easier for the molecular people, right? It's easier to just go online to see, has someone else done this for me? Do I have to request a loan to do this? And for me, I'm really slow about publishing. It's one of my greatest flaws. And I having a repository where data can be stored that I can share with my collaborators makes it easier. That has that you can place a moratorium on, that it makes it easier to be able to feel safe about your data's security and collaborate with colleagues and not have to worry about someone coming along and stealing what you're doing if you're very protective that way. But it also allows collegiality. Morphosource is particularly nice because one of the things about it is that people can contact, they can see what you've done. They can contact you and ask you, can I use this scan? And then you can sort of suss out, like, what are you doing? Because if somebody's doing something totally different for me, I'm not a developmental biologist. If somebody's doing something with development and they just need an ontological series, I don't care, right? So it's not, to me, it helps a lot. So.
On the topic of having things online and more easily accessible, one of the questions was, it's often stated that the increase in digitization means increased access, which will probably mean there's going to be more diverse individuals that can now have access to your collections. In your opinion, to what extent is this statement correct? Do you feel like there's been more people, specifically more Black people, able to get access to collections and or use this for science because of digitalizations? And if not, what steps do we need to take?
Corey, you want to respond to that? And I have something to say about it too. Oh, Corey just said goodbye. So actually that last question kind of touches on this issue of decolonization, doesn't it? I mean, the tradition in natural history collections is that, they're kind of like these the perception is that they're these old dusty cabinets, you know, that are stuffed with specimens. The specimens have to be pulled out of drawers of cabinets and whatnot. And, you know, this has been true to a certain extent. Once you digitize them and make those digital analogs of specimens available, you know, who doesn't want to put that online and give it access to everyone? And, you know, once it goes global, yeah, that naturally does give people access to these objects that would otherwise have to travel to the museums to study them. So I think this is a great opportunity, and it does help in the particular cases that we talk about with making cultural artifacts from countries available to people from those countries, but it also helps broadly and generally to give people access to these specimens.
Yeah, and I just want to jump in and agree, of course, with you, Hank, that the more that individuals who aren't scientists or who don't have that particular specialty can access information, all the better. And if we work to put materials and images into a learning context, you know, shaped for a particular audience. And so those are some of the things, you know, I've been doing at the UCMP with this intentional inclusion in modules that we know, will be shared with two-year college audiences or a certain demographic, and shaped in a way where they hopefully can relate a bit more to what we do. And, I can't help but mention also just all the barriers that are inherent in field work. And so some of what's been at least addressed over this last year and a half is making what we do in the field, more accessible to others. So it's, even before we initiated these virtual field experiences for the EPIC project, we're seeing increasing numbers of remote field experiences that used all of the, technology available to us now. And we think of places where we collect, many of these places are not accessible to everyone or not accessible anymore. And half the time we don't want everyone going there anyway, but, there has to be, I think, more intentional ways of capturing, where these places are, that we get data and why there. So.
And just a second question that I personally had, going back to the education on digitization. In my experience, when I was using NCBI for my research, it took numerous classes to kind of understand what NCBI was doing and get a phylogenetic tree. My freshman self did not enjoy it. A lot of people stopped worrying about that, but I had questions that required NCBI, so I eventually got comfortable and really liked it. Are there, lessons out there for other undergrads or maybe even high school students to easily say, I know what this is doing, what questions can I ask? Or is that something that needs to be worked on? I know there's general PDFs for scientists on how to use iDig or how to use NCBI, but is there a need for better educational resources for undergrads and or high schoolers?
Lisa, you have some comments about that?
Well, when we put together the modules that I highlighted for the virtual field project, there are some parts of the modules that do highlight the technology. It was more focused on the gigapan photography and the capturing of images in the field. But we try just all along the way to provide links and resources and send people to other museum pages where they can drill down a little bit into what scanning and digitization and analysis of all that is all about. Yep.
Yeah, I'll say from my experience that, I teach at pretty high levels, upper levels and grad student classes at Tulane University. So I don't get to interact with freshmen in the past, in situations where I've worked with high school students or even elementary school students. I was always challenged, about how to present the material tool. But one thing that I noticed, even with kindergartners, they are fascinated by specimens. And, you know, they just take such an interest in it. You could just see it, you know, in their eyes and their excitement. So, yeah, I think that we all as professionals have to figure out how to get really good at how we present this information to them and get them to capture that excitement so that they want to study these organisms because I think there is a lot of interest there.
And then I think we have a question from Adenia. Do you want to go ahead and ask that?
Yep, I will. And I see the person in the chat, somebody else has a question that follows mine, which actually is related to mine and to yours. And I think it's a question that a lot of us have in this community. And so when I say in this community, people who are Black and Naturalist museums. And so my work, part of my PhD stems in terms of looking at broadening representation in STEM. And one of the first things I was doing for that was looking to see, like, how are we using collections for education? And so that's kind of going off of what we were talking about before. And I know, Hank, you're saying like this in your forte, but I'm just thinking generally, right? Because Lisa, like what you're doing a lot at UCMP, but we have a lot of different museums across the globe, and I don't think we're all doing a lot equally to some extent. And we've also not been doing a lot for the same period of time. And so this question spans to like natural history museums as a whole. The person in the chat that comes after me basically kind of asking my question, but the main point of it is, so they said, what can we do to engage more Black students and scientists with specimens? Making data available online is likely not enough. And so I mentioned that here because my question is literally folded into that. And so, yes, it's more accessible, right? We put it online. You guys give some really good example. You all, forget the guy's spot, I need to correct my language there. You all give really good examples of how they're more accessible, especially for research. And it's really nice to see Black folks using it for research. But there's a whole huge group of Black people and non-Black people of color who have no idea about collections, even though they're going virtual, even though they're online. And so my question is, as museums as a whole, as an entity, natural history museums, do you all have suggestions towards across the US, across globally, how we can work collectively to not just digitize and make them accessible, but sort of intentionally ensure that they're getting to people who should be aware of them and people who like, they don't have the access to journals because they don't have the whole behind the scenes paywall. They don't have access to journals. They're not necessarily going to conferences. They're the public who we keep saying we're doing stuff for that have no idea what we're doing. So it's a very loaded question. I gave a lot of kind of overview around it. And I would love to hear, even if you don't do stuff, like in a perfect world, if you can tell a museum, we need this, we're gonna do this, or work at a museum who has education, whether it's virtual, whether it's a university-based museum, non-university museum, whatever it might be. How can we, I want to dream build a little bit, like what would that look like? What are your thoughts?
So Lisa, I'd like you to start with that since you're
Okay.
working outreach and education, but I have something to say too.
All right. Well, it is true. Just because we make what we think are the best, you know, the most awesome, unique resources from these digital products, it doesn't mean, you know, everyone is going to come. And especially the audiences, the Black audiences that, you know, we hope do connect with our resources. So yeah, the word is being more intentional, and it means we have to expand our networks. We do tend to go to the same kinds of conferences where, you know, other folks that work at museums are there. But you know, I've been exploring some different funding opportunities, and the University of California does have a funding opportunity that is that's related to the HBCUs, and so partnering with HBCUs to support STEM education, and I've also been working just with my other specialty with the ocean sciences community on a series of workshops around climate change and climate change education and impacts. And in the most recent workshop we're organizing, one is required to come. If they apply to come, they have to apply with a partner who is at a university completely different from theirs if they're at an R1. So that's a great opportunity to invite someone at a Hispanic serving, minority serving, or HBCU. So we do have to get, out of our boxes and silos, but we also have to seek, funding to do the kind of work we would like, because you can't just ask somebody to be involved or assume they'll be excited about our resources without some context. So that's just something to think about.
Yeah, and what I was going to talk about was, at the times in the past when I've worked with students, kind of at-risk students at underserved schools, or whether it was high school level, elementary levels, what I found was important is to work with teachers. So I didn't really know how to begin to present the content, to them. Like if we were looking at a skeleton of a fish or something like that, a cleared and stained specimen like Corey talked about. And I didn't really know how to present the information other than just in technical terms about how we produce the specimen and how people use it. But the teachers, they know how to, to break it down to the level of their students. They know the kind of things the students can capture. I think there's lots of opportunities for us to take things like CT scan, segmented bones in CT scans, compare that to a specimen like a skeleton of a fish, and point out those same bones. I mean, who hasn't eaten fish and was picking meat off the bones? I know I did when I was little. So I mean, there's lots of ways you can relate, to a fish that you're eating as a meal, and skeletal elements in that specimen. So I just think that we have to work with educators, and we have to get creative about how we present the content to these younger audiences, these underserved communities, and then use that as a hook for trying to get them interested in it and pursuing that as a professional, or even just to go to college and learn more about science or biology. Skip, you have anything to add to that?
I mean, yeah, I'm speaking at this from the angle of not being directly involved with outreach at beyond college at the moment. But I will say that there is a general not just in minorities, but I say minorities are particularly afflicted by this. There's a general underappreciation. And like, if I'm going to go do science, why should I do anything that isn't going to be financially lucrative, right? So it's this inclination to be like pre-med, right? So I feel like a lot of people are inclined to think that we can fix this problem by addressing it at the college level. And at that point, I think it's kind of too late. So you can lead a horse to water, but they're not going to drink, right? So I think that, again, like when I was at Berkeley, I was involved with BASIS. So reaching out to little kids, and teaching them about like bringing skulls to the classroom and stuff like that. So getting them more inclined to understand the appreciation of natural history museums and understand that, like nature isn't disgusting or terrifying at a young age is, I think, the key. And I just think that that's something that, frankly, I don't think we're going to really see the benefits from, not in our lifetimes, right? I think this is going to be something we're playing the long game. It's like sowing the seeds for the kids now, but for most of it, like I'm teaching college kids now, and they're just like, why are we talking about dog genetics in the intro bio class? Not understanding that they have DNA too. So it's just like, what? So, there's nothing we can do for these people at this point. I realize it's kind of nihilistic, but I think that's the realistic aspect of it. So it's just like, for people who are disinclined to see the value of CT scans or why that might be interesting, now I think that getting them while they're young is the key. So if that makes sense.
I think it does make sense. So how many of us are in this for the money? Raise your hand. I don't see any hands now. We do what we do because we're passionate about it. It was something that maybe we were excited about at a young age, like those little kids I was talking about working with. But, that's a passion that stuck with us, and it's driving us now, to study yourself. Now, I think that I think to a certain extent, we have to change people and people's ambitions. You know, look, there's always going to be opportunities. People make a lot of money in this country. And we know that it's a very small segment of the population that can be wealthy like that. But there's lots of opportunities in more mid-level positions, you know, for people to have careers as scientists, to have careers as teachers, to have careers like this where, you know, people who really love that particular vocation can enjoy themselves. And I just think that we need to emphasize that more. And the other point that I'd like to make is that we need to convince more people to be more concerned about the planet and what we're doing to it. It's the only way we're going to reverse some of the horrible things that are happening right now. I don't know how to do that, but you know, it's just something I feel is very important for that, whatever segment of the population. Hopefully, you know, a lot of the minority and underserved community, we can get excited about trying to save the planet, and that involves, you know, paying more attention to the creatures we share the planet with, the environments that they live in, and so on and so forth. That's my soapbox of the day.
Yeah, and Hank, given that climate change is literally at our doorstep, I do feel like, you know, we've turned a communication corner there with a lot more sense and awareness of just how much, you know, the planet is different from even last year and the year before. So that is more attention getting, but at the same time, people and families have such immediate concerns, you know, it's hard to maybe make the kind of investment that we know scientists have to in work and research to, you know, do work that's connected to those topics. But yeah, clearly there's work to be done at the elementary level on up through K through 12 and in college. But yeah, I know what you mean, Skip. By the time we get them, okay, at the university, it's just really hard to sell somebody sometimes on our disciplines.
Yeah, I think part of it too is that, I mean, I also don't know if there's a solution to this. And I think that our current political situation has highlighted that there probably isn't, not an immediate one anyway, that there is a general undervaluing of just knowledge for the sake of knowledge, because that's what we do, right? Like it's curiosity is not something that's immediately rewarded. And I think that there is an incentive to be incurious in general. People really take pride in their incuriosity, right? So especially in our nation, we live in a nation of know nothings. And I think that if you come from nothing, that there's very little, especially when it's just like, how much money do you make, right? Like it's, you know, like I talk to my mother and she knows what I do, and she doesn't have a college education, but she's like, so why would anybody give you money to study snake skulls, you know? And it's just like, I'm just like, because it's interesting, right? Like that in and of itself should be an answer. And I don't think that if you, like, I don't know how to convince somebody that something is interesting if they don't already find it's interesting by exhausting the means that we can already give them, right? So I don't know if that makes sense, but yeah.
Yeah, it does make sense. And I don't mean to paint this as a, you know, a challenge that there's an easy solution to, but, you know, I'm just saying that it'd be nice if we can get the students when they're young enough to get them really interested and passionate about science in general.
I 100% agree with that. I 100% agree with that, yeah.
So everyone, I have to run, but it's been such a pleasure to participate in this panel.
It
And
really has.
Mhm.
Yeah, for me too.
Thanks, Lisa. Thanks for joining. And before we wrap up, one quick thing to this question that I want to note is that as museums, we typically, like as being part of IDIC Bio and studying, like, you know, who's using collections, we really are lacking demographic data in terms of who's actually accessing it. So I think that's one thing we can look at as museums as a whole. And then if we have some baseline, we can look at how it's changing and whether that is starting at K through 12 or undergraduates. And I think to the points mentioned before, and we need to get people at every level, because right now the undergraduates are moving into jobs faster than the K-12 folks. And we need people at every step of the way. And so I think I really appreciate the conversation you're all talking about, like the reality of it. It's not a simple, like, let's do this and this will happen and then we move forward. It literally is something that we need to keep asking questions about and asking about how we can do better, because it's not just I want to dream it, so make it happen. And so I think if nothing else, just has illuminated that aspect of the need to really be intentional of how we think about collections, now we think about accessibility, now we think about inclusion, if we really want to diversify, thinking about the folks who are often most marginalized in our spaces, given the climate that we are in the US and across the globe, essentially. So thank you all. I hope that makes sense, that aspect.
It does. And thanks for the helpers, the chat monitor and the moderator. And well, I guess I'll see you tomorrow for day 6.
Great.
Much appreciated, everyone.
Bye.
Take care. Bye-bye.
All right.